So if everybody is okay with recording, thank you for everybody for joining. My name is Andrea Zapp and I'm with Aquatic Sensors. I've got Patrick Sanders if you want to give a wave. He's from Aquatic Sensors as well. Just a bit of housekeeping. If everybody could mute yourself for about the first 20 minutes while Andrew goes ahead and he'll do some technology talk and a bit of demos. And also if you could put your screen into speaker mode and that will allow you to see all the demos and what Andrew has to show us for today because he's got some good stuff for us. Okay, so I think I'm recorded. Can everybody, Patrick, can you double check I'm recording this? Everybody okay?
Yes, you are
Perfect.
recording, yes.
Yeah, you are.
Awesome. Awesome. Okay, so let me introduce Andrew. Andrew Monk is an Oxford-educated CEO who excels in growing businesses that create innovative products and services in the technology sector. In 2014, Andrew founded ioLight Limited with Richard Williams, and the company filed a patent for the world's first portable digital microscope with 1 micron resolution and the ability to share images. And this is when the magnificent mobile microscope was born. And in his spare time, Andrew can be found playing his bass guitar, which we were just talking about earlier. So I will pass it over to Andrew. And for those of you who I forgot to mention, we are here to talk about ioLight's technology. So over to you, Andrew.
Thank you, Andrea, and thank you, Patrick, for having me. And good morning, good afternoon, everybody. It's very nice to meet you. So I hope you don't mind if I go very slowly because I've had a long day. It's 6 o'clock in the evening here, so that's fine. But we've had a beautiful California sunny day today, so at least we've got that sorted out. So what I'm going to do is I'm going to go through some slides. I hope Andrea is going to keep me straight on timing. I'm going to go through some slides. Do interrupt, please, if you've got questions. Is that okay, Andrea, if they interrupt, or would you want to? Yeah.
Yeah, if you're happy with that, we're
well, I'm happy. That just makes it less boring. And then at the end, we'll have then I'll do a demo and I'll show you some stuff, and then we'll have a question and answer session at the end, and we're aiming to finish in an hour's time. So that should be good. Okay, so let's see if my slides go. Yep. Is that okay, Andrea? Can you see that?
Yep, you are good. We can see that.
Good. Okay, cool. So this is what we're doing. Everybody knows about disruption. It's the buzzword in tech startups. We're disrupting this and we're disrupting that. We are disrupting cell imaging. And what do I mean by that? People have been looking at people have been looking at cells for 400 years. So what do I mean by disrupting cell imaging? We'll have a look at that. Now, as Andrea said, Richard and I founded the company in 2014. We're located in Hampshire, right in the middle of the south coast of the UK there. Those of you who know England, you might know Southampton and Winchester. Those are two cities that we're very near. And we're also very near the Isle of Wight and the New Forest, the New Forest is very famous because it's not new and it hasn't got any trees. So that's a very English bit of a history there. Nelson chopped them all down to fight the French. So the product was first launched, our first microscope was launched in 2016, and we've sold well over 700 units now globally, all the way around the world. But the thing I wanted to say to you is that more than half of those have been sold in North America. So this is not a little British company that sells the odd unit to North America. North America is our core market. In fact, I can tell you that we've sold quite a few since the beginning of our financial year on the 1st of April. We've sold quite a few microscopes, but we haven't sold one in the UK yet this year. And we go to a wide range of industries, but our biggest industry is the environmental industry. And I'm guessing that a lot of you are interested in HAB identification, and we can talk about that later too. So that's a bit of a history lesson. So we're disrupting cell imaging. What's wrong with microscopes? Most of you have used microscopes. I love microscopes, beautiful, beautiful images. And this product, like the one that this scientist is using here, has developed over 400 years. But the compound microscope was developed very early in that history. And that design that is being used there is probably 350 years old, the basic principle. And there's not much that's changed about. I mean, we've got electron microscopes and we've got some techniques, but the basic compound microscope there hasn't changed very much. And they use a lot of expensive bench space. Those of you who work in labs know that bench space is very expensive. It seems like a weird thing to say. It's just a table, but benches typically are in air conditioned and filtered clean rooms, and real estate like that is pretty expensive. And then there's this strange thing that's happening at the moment with pandemics, with COVID, and with environmental science. That all happens out in the field. That doesn't happen in the lab. I've never seen a lab with a harmful algal bloom in it. And those happen out in the lakes. So more and more science is driving researchers out into the field so that we can go and look at these things. In COVID, the last thing you want is hundreds of people who might have COVID coming to a central test place. That's a disaster. You need to go out to them to test them where they are. And in America, England, and Canada, that's not too hard to do. But in Africa and India, it can be a lot harder. Then there's another thing that we've discovered really quite recently, and there's going to be more published on this very soon. And that is we're finding that shipping samples, particularly harmful algal bloom samples, to the lab damages the sample. And that means that you are not going to get the same result from a sample that has been shipped through the post or driven in your car to the lab as you will do from a sample that's tested at the lake. The other thing with traditional microscopes is you need to know how to use them. It's quite funny when we train people on our microscopes, people say, oh, I must be careful not to do this, and I mustn't touch that, and I mustn't drive the lens into the slide. We've only ever broken 2 microscopes. One was at a show when a 4-year-old picked it up and started using it like a hammer. And that wasn't too bad. We just straightened that out and it was fine. And the other was a veterinarian who dropped it into a bucket of what she said was water, but it was more corrosive than that. And she'd been with horses all day. So I suspect that would come from a horse, not from a faucet. So they're pretty robust, our microscopes, and you can't take a microscope like this out into the field. You can't take a traditional microscope into the field. And you definitely can't. I know, America, Canada, huge countries, and you guys travel a lot on airplanes. You can fly with a compound microscope, but if anyone's ever tried it, it's really not easy. Whereas I quite often fly with 3 or 4 of our little microscopes in my briefcase. And the other thing with microscopes is it's an optical device, so the interpretation is as good as the operator. So some people will have different opinions. What we'd like to do is to connect all microscopes to AI so that we've got a consistent interpretation of every image. And those of you who've tried to connect optical microscopes to the internet will know it's actually very difficult. A lot of microscope software is compatible with Windows XP and old obsolete versions of Windows, and you have to hook it up, and it's just a pain. Whereas our microscopes are internet native. So here's the solution. Here's one of our little microscopes, and you can see an iPhone there, which shows you the rough scale. It fits in a jacket pocket or a backpack. It weighs a pound. They're really small. And we lose some resolution doing that. And don't ever, I always get worried when people compare the resolution of our microscopes with an optical microscope, because ours are not as good, but it's a trade-off. If you want the very best image you can possibly get, then you need an optical microscope. but our microscope can do an awful lot, and it's internet connected, so we can do a lot of the interpretation. The display is on a cell phone or a tablet or a laptop, but people tend to use cell phones because they're so nice and portable, and you've got them with you anyway. Modern cell phones have just got brilliant screens. I've recently got a Google Pixel 7a, and it's just fantastic. The screen on it is really good. And of course, once you've got the image on your phone, you can just share it any way you like. You can share it using your corporate internet. You can even share it on Instagram or Twitter. Yeah, you can. Sharing images from an ioLight microscope is just so easy. And more importantly, you can plug it into apps, and we'll look at one app in particular later on. The battery lasts all day if you're working in a normal way, and if you want to have it running all day permanently with a video display, say you're at an exhibition, a show on your booth or something, you could just use a backup battery pack that you would use with your cell phone. Best of all, these are simple to use, and that means we can get kids involved in environmental things, and that's just so important to get kids to learn not to break the world the way that my generation has broken the world. And many of our citizen science projects that we do have 3 generations of people in a family working on them. There's one group in particular in Wisconsin where the granddad and the granddaughter go out looking for cyanobacteria, and it's so great to see those multi-generational things. Then at the other end of the scale, we're doing real science with AI-powered auto-diagnosis, and this is going to lead to much better data, big data sets. And I think my dream is that this will start to take over just because of the amount of data we're generating, because anyone can generate data. You don't have to have a PhD to generate data with one of these things. So Patrick and Andrea, I want to say something about Patrick and Andrea. Thank you guys for giving me this opportunity to say hi to your customers. It's a real privilege, and working with Patrick and Andrea at Aquatic Sensors over the last couple of years has been an absolute joy. They really understand these products. It's not just, some of our distributors it's in a catalog with 1,000 other products, and they have to try and remember what it is. Patrick and Andrea know these things backwards, forwards, and inside out. And although their home zone is water quality, and that's where their deep expertise lies, they're also getting to know some of our lab products, and they're stretching out into the lab world. So thank you very much, guys, for all your hard work, and it's been a real pleasure working with you, and let's hope that goes on for many years to come. So Patrick asked me to put this picture of our product range in here. So we basically have 2 different magnifications of these folding microscopes that you saw on the left-hand side of your screen, and that's a 1mm and a 2mm field of view microscope. And we'll have a look at those in a minute, and you'll be able to understand the difference between them. And then if you notice on the microscope on the left here, that is slightly different. It has an XY stage on it, and people were using our microscopes to count cells, and it's quite difficult just sort of trying to find the right place on the slide. So we've patented this XY stage that you can move independently in 2 directions, in the X direction, the Y direction. And if you're doing some cell counting, if you're counting cyanobacteria cells, for example, or if you're a veterinarian and you're counting parasites, being able to count in straight lines, being able to go up and down the lines on a counting chamber is really important. So that there's a 1mm XY and there's a 2mm XY. And then we went a bit crazy a couple of years ago and invented a fun product because we can. And that's this one over here with the F on it. And this is the world's only pocket fluorescence microscope. And this is generates 465 nanometer excitation. And that will excite fluorescence molecules in GFP, YFP, and a number of other fluorescent molecules. And most importantly, this can be of use when you're looking at chlorophyll A and phycocyanin and some of those interesting comparisons like that. it doesn't, it's a very weird product. It doesn't do everything because there are a lot of compromises in such a tiny microscope. But the good news is we also have a large laboratory microscope like this, which also has fluorescence in it. And so if you're doing serious fluorescence work, that's the tool you need. So that's our cell imager, and we'll have a look at that in a minute as well. And that's available either with or without fluorescence illumination. So I mentioned 1 millimeter and 2 millimeter field of view. And so the optical magnification on our microscopes is fixed, but the resolution is so high that you can zoom in and you can do the digital zoom and you can see some, quite good detail. As I've said before, and I'll keep saying, these images are not as good as a well set up optical microscope. You're making trade-offs. If you want to look at E. coli or something like that, you need an optical microscope. One day maybe, but there's a lot of compromises in these products. So the 1 millimeter field of view, and what we mean by 1 meter is from the right hand edge of this image to the left hand edge of this image is always 1 millimeter. And that's great because you've got a scale bar built in. You can measure the width of the image, and then you can divide any measurement you make on the screen by the width of the image, and you've instantly got a scale there. And you can see that with 1 millimeter field of view, that was the first product we made. It's got the highest magnification that we can easily do using a cell phone lens. And you can see with this wing, this is a housefly wing, and you can see the little spines on the side of the wing. I can tell you, because I know that's a 1 millimeter wide image, those spines here, they're about 30 microns long, and they're about 4 or 5 microns in diameter. So they're quite small. And you can see them very clearly in that image. Obviously, the things you can see are the little tiny hairs on the wings that are much smaller than that. So that's not the smallest thing we can see. And then, as sometimes happens, very soon after we launch that product, some of our customers say, "Oh, it's too powerful. We want to look at a big field. We want to count. Gastrointestinal nematodes in a fecal sample, for example, for veterinarians." And they needed to count across a whole area. So we built a less powerful version, which you can see on the right. This is 2 millimeter field of view, and you get a much bigger field of view. You get better illumination. So for opaque samples, you get better incident illumination. So for samples that are opaque, like leaves and coins and things like that, this microscope is a better bet than the more powerful 1 millimeter. And again, if you've got any doubts, talk to Patrick and Andrea, and they will talk you through your application, and we'll figure out which is the best one. So the 1 millimeter is great. If you're looking at cells, you're going to need a 1 millimeter, the most powerful microscope around. If you're counting, you'll need the 2 millimeter. And if you want to use the Bloom Optics AI app that we'll talk about in a minute, you need the 2 millimeter because their library is built around the 2 millimeter microscope. They're looking for colonies of HABs, not individual HAB cells. So that's why they want the big view, not the in tight view. So in case there's anyone here who's not from the HAB world and is not familiar with HABs, HAB stands for harmful algal blooms. And it's a real portmanteau of terms. It covers all sorts of species and genera of things. But basically, these are brightly colored slicks that sit on top of water, and that can be the ocean or the lake or river, and under certain circumstances can be toxic. And they can be very toxic indeed. I was talking to a toxin expert the other day, and he told me that milliliter for milliliter, microcystin is as toxic as cobra venom. So it is very toxic indeed. Thankfully, when you put 1 drop of cobra venom into a big lake, it's not as bad as it sounds. But you don't want to be in a lake that's got microcystin in it. So the toxins, different toxins do different things, but typically symptoms would be rashes, diarrhea. Sorry about the weird British spelling there. and death, which is not a good side effect. So there are two impacts of this. One is public health. Obviously, you don't want people anywhere near water, people or pets or livestock anywhere near water that has harmful algal blooms in it. And there's also an economic impact on businesses by the lake. So there's a commercial risk as well. The standard solution to that is to send the sample to the lab, and that takes typically 5 days to turn it around. We can, using the Bloom Optics app, we can analyze it in 5 minutes. And that means you can take action immediately. You can close the lake or you can open it up knowing that it's safe. So our lovely partners, some of you will have met Igor Mioden, who did one of these webinars a few weeks ago. They're based out of Utica in New York. They have a machine learning app. So they've built a library on the web of 20,000 images. It takes the image that you've taken on your ioLight microscope, it uploads it to the web, it compares it with the images they have on the web, and it gives you, it identifies 6 cyanobacteria genera. And we're looking at a genus level, not a species level, because if you've got microcystis in your lake, you need to be worried irrespective of which species it actually is. So that's the philosophy. And they've also developed an algorithm that counts the cells, so they can tell you what the cell count is, and you can compare that with state or EPA thresholds. And this reporting from the ioLight photograph image you've generated takes about a minute. And we've just developed this microscope for laboratories too. This is the same microscope but turned upside down. So the camera is on the bottom underneath that flask, and the illuminator is on the top. And this is to monitor cell lines. We all know that people do very little experimenting on animals now for new medicines and therapies. It's done on typically human cell lines that are grown for the purpose inside an incubator that simulates the human body. So it's at 98 degrees F. It's humid. It's full of carbon dioxide. And when you want to look at the cells, you have to take the sample out of the incubator, and that disturbs the sample. It changes the conditions. It introduces contaminants, and our microscope can live inside the incubator, and it doesn't. You don't mess up your sample when you take it out, and you can watch them growing. You can look for infections. You can check for convergence. And we've also on this cell imaging microscope got a few really cool features. So that includes fluorescence, as I mentioned before, and you can see on that microscope, the blue light underneath, that's illuminating those GFP cells in this particular sample, stained sample. It also has dark field, which the folding microscope doesn't. It has a high contrast, and for the optics people among you, that's a low NA mode. It's like adjusting your condenser on your conventional microscope, and that's ideal for transparent cells in water. This microscope also has digital phase contrast. We can simulate phase contrast using our electronics, and it has time lapse and Z-stack, and all of that on a little compact electronic microscope. And this is a video of some human kidney cells that are being grown in a lab at the University of Southampton, very near to where we are. And this is a HEK293 as a typical cell line. And this image, this is a time-lapse image. It's taken one picture every 10 minutes for 48 hours. And if I talk a bit, you can just now start to see the cells starting to grow, and the area of the image that they're taking over now, they're starting to converge. You're starting to see the cells stick together. And it's really quite interesting in doing them. And all of that can be done inside a microscope with the door, inside an incubator with the door shut, so there's no contamination. So what about the future? Well, the currently AI program works on cyanobacteria, so it's freshwater AI only. But we're working currently with Bloom Optics to build a database for marine HABs. So we're looking for Carina brevis, red tide, for diatoms, for dinoflagellates. We've already done a lot of work in the UK on Alexandrium minutum, and CFAS, which is a government marine health agency in the UK, is very keen to prevent paralytic shellfish poisoning. in people who like me who like shellfish. And in Europe, we eat quite a lot of mussels and oysters and crabs and lobsters. I know in the Northeast, you guys do as well. So getting Alexandrium anywhere near shellfish beds is a disaster. And so we work with CFAS to protect them against those HABs. And this is a bit of a big statement, diagnose anything anywhere. What I'm saying there is, in principle, once we've trained the AI, we could detect anything. And this image on the right is a cell line. It's another human kidney cell line. And we're isolating those cells and watching individual cells grow there. And so one day we could automate cell lines. We could get send you a text when the cells have converged or whatever. So I think that's an exciting future. Also, I think tropical disease is a very important one. Schistosomiasis and other parasitic diseases like that are big killers. There are 15 million people a year treated for schistosomiasis, and it's a really nasty disease. And this microscope could help to diagnose it automatically. In the hands of a technician, you wouldn't need to be a trained medic. And that question at the bottom, I will leave you with. I think in some applications, an electronic microscope plus AI in some applications might be better than a compound microscope. I don't think the images are ever going to be better than a compound microscope, but the data might be. Cool. Were there any questions from that spring to mind?
If anybody wants to come on and ask Andrew a question at this point before he gets into a demo, feel free to put it in chat.
Okay, we could crack on and just have a look at one, shall we? I mean, we're going to come back to Q&A.
Yep, put it. Yeah, it'd be great to actually get a visual on your technology.
Oh, my technology on my hands. I've just been making my supper, and we're having beetroot for supper. So my apologies, and my hands are all red. I suddenly thought while I was peeling this beetroot, this is not a good idea. But here you go. Right. So this is our microscope. As Andrew said, I'm a bass player, so my left hand is a bit bigger than some people's left hand is, because bass players' left hands get stretched. But it's still a very, very small microscope. And we wanted it that way because the idea was that, you carry your cell phone everywhere, and we'd like you to carry this everywhere as well. So, and we wanted it to be flat. The problem with that is you need an optical system up here to observe the sample. The sample sits on a stage so that it doesn't move. We don't want the sample moving around when you're imaging it, because you'll get a terrible image. And there are some portable microscopes that don't have stages. I would steer clear of those. So to get the optical system up here, this has to fold, and it took us 6 months to figure that out and to patent it. So that's what our patent is. And then the sample just goes on the stage like this. And then the image is on your cell phone. I'm going to use an iPad here just because the screen's a bit bigger and you can see it. But most of our customers use their personal cell phones. So I'm just going to connect to the device, and you might have seen the illuminator come on, the bottom illuminator come on. And just straight away, we've got an image, and it's that simple. That's how easy it is to connect. I'm just going to focus down on the sample and start to look for the normal bits of dust and things on the slide. There we go. And now I'm just going to, using my fingers, because this is not an XY microscope, I'm just going to start wandering around the slide to see if, oh, there's something. Now this is typical, you see. I'm showing you this slide, and the previous occupant of this slide has obviously gone to the bar. I promise you there is something on this slide somewhere. There we go. And now I'm just going to focus it in a bit better. So you can get quite close just by hand, but you don't need to worry about that. Obviously, I've done that a few thousand times before. Because on the display here, there is a fine tuner, and this is actually moving the lens. So we can pick out any feature we like in the depth of field of this. And some of you who can see that very well, I know it's difficult, you may notice that's an aphid. So it's a green fly or something like that. And we can just tap on the camera here, and that records what you were looking at before is a video, but now this is a 5 megapixel high resolution image that's saved on the camera roll, and we can now just share that using any app, any image sharing app. Very, very simple. There's I don't want to take up too much time unless there's specific questions. There's also a video camera here. If anybody likes recording video, for example, if you're looking at Alexandrium, the way that Alexandrium moves is one of the important things to identify it. And you need a video of it, not a still. And this is one of the best little video cameras, video microscopes out there. It's, as far as I know, one of the few pocket video microscopes on the market. And we have control over the illuminator here. I can turn the illuminator off. I think you know what's going to happen. And we can turn it on to super bright. It just changes the contrast a little bit because the camera will shut down. And that's just a slightly more washed out image than the one, didn't change, than the one I was looking at. Yeah, it's slightly better contrast here. We've also got an illuminator at the top above the sample, which we can turn on. It's not going to make this image look any better, but for some samples, like for example insects, it's a real help, that top illuminator. And we've also got a settings menu here, and I can overlay you may be able to see that we can overlay a grid here of 10 squares, and that grid is programmable. You can change the width of those squares to match the microscope. So you've got 10 squares for a 1 millimeter microscope and 20 squares for a 2 millimeter microscope. So you can make the squares equal some sensible amount, or you can set them for any random value you like. So we can also go to the stored images, and this is just my standard image. It's got a bit glary. Yeah, that's not great, but that's my standard image gallery in the iPad, and you can see the photos I've been taking there, and you could just share those as you normally do with any image. And we just open the app again, connect, and there we go. Cool. So I think now's the time for me to stop talking, isn't it, Andrea?
Oh.
I think you're on mute, Andrea. Hey, Andrea, you're on mute.
Oh my goodness, sorry about that. That was my trick of the day. I was going to ask if you had your inverted cell imager also. You wanted to show us the size of that one, or are you saving that for grand finale?
No, we can have a look at that. And so let me, while we're down here, so the cell imager, so this microscope, the portable one, the camera is at the top and the illuminator is underneath, and we're shining light through the sample into the camera. This one is inverted. So the camera is in the body, it's upside down, and the illuminator is at the top. So we've got more space, so we can do more sophisticated things. It's significantly bigger, but it's still really small. This one weighs less than a pound, and the bigger one weighs about 4 pounds. So things like, some people find this hard to learn to focus. Here we've got a nice big focusing knob, so that you can actually manually focus. It's very much easier to work with. So if you're on a boat or in a lab that's by a lake or in the field, this might be a better bet for you if you don't actually need to carry it every day. It's a little bit more expensive, but you do get some benefits. You get slightly better contrast on this. Also, you get the fluorescence feature, which I'm not going to show now unless anybody's desperate to see it, but I'm happy to arrange another demo of that at another day. The other thing we can have a look at is I mentioned the XY stage. This is an XY microscope. You get out of the way. Stealing all the limelight. This is an XY microscope, and it has a stage that's slightly different. This is a metal stage, so the microscope's completely different. You can't retrofit the XY stage to the standard microscope. But it works exactly the same way. The stage is steel, and it's got railroad tracks in it and a window in the middle. And the stage clips on very nicely using magnets, and the slide goes onto the stage like that. So now we can move the whole stage backwards and forwards in the Y direction, and we can move the slide itself backwards and forwards in the X direction. And so we've got controlled movement with X and Y being completely independent. So that's the basic family. And there are 1mm and 2mm versions of the folding microscope, and there are fluorescence versions of both the inverted microscope and the folding microscope.
Awesome. Thanks, Andrew.
Cool.
Does anybody have any questions from earlier? Any questions about the microscopes? Any questions about the bloom optics? All right. So we have. So one question is, I'd like to identify plankton in the field for HAB species. Which microscope would be best for this task? And what are the challenges I might be facing using this microscope?
Okay, so there are two ways of doing that. If you are an experienced taxonomist and you know your HABs, you're going to want to use the 1mm microscope because that's the most powerful microscope, and it will give you the closest in view. Now, of course, people are getting very interested in nano HABs at the moment, and these very tiny HABs. Some of those are too small to see even on a bench microscope, and they'll definitely be too small for us. And then going up the range, if you're interested in cyanobacteria, you should consider the Bloom Optics app, which automatically identifies cyanobacteria from the shape of the colony, not the shape of the cell, the shape of the colony. And for that to be compatible with the Bloom Optics library, you need to have the 2mm microscope. So 2mm microscope, if you want to use the AI with Bloom Optics, and 1mm microscope if you just want to do it by eye yourself. The challenges are pretty much the same as you would see on a lab microscope. In fact, you have slightly fewer challenges because, as I mentioned earlier, there's a lot of emerging evidence that transporting cyanobacteria cells from a lake to a lab, if you're going to air freight it or whatever, courier it or drive it in your own vehicle, what we're seeing is a lot of the cells get lysed on the way, so they get busted open. And so what you see in the lab is not the same as what you see at the lake. And so in some ways, the ioLight microscope is going to make that easier for you. And Patrick just put up the list of 6 species. Thank you for doing that, Patrick. I can never remember them or pronounce them. So I'm very grateful to you for flashing up the list of 6 species that are 6 genera, that are automatically identified by the Bloom Optics AI. Of course, if you're an experienced taxonomist, you'll recognize many, many more different species within each genus.
And just to talk further on that, they're always expanding the library, right? So every year, the library gets bigger, more than 20,000 pictures. So hopefully we have more, and we can customize it for certain genuses. If anybody wants to make that library bigger, we're more than happy to work with you.
And a good example of that is the work that we're currently doing with New York Sea Grant and New York DEP on identifying marine HABs. Obviously, marine HABs are much more varied and typically smaller, and they're motile as well. So that brings a whole new range of problems. But we're currently building a database of marine HABs, and anyone who'd like to know more about that, we can set up a separate call for you. And there's a question from Vanny there, Andrea,
yes.
about the slides. Yeah, so the slides are absolutely standard glass slides. So if you're going to use the portable microscope, this one, there have to be microscope slides. They can be either the flat ones, or we use a lot of cavity slides, the ones with the little dip in. And for cyanobacteria, that's the best way of doing them. You just prepare a couple of drops of your dip sample, grab sample into the cavity, and that shows you what you need to do. Yes,
Further to that,
you ask a question about volume. Yeah, we've done some clever sums on the volume. That's a very good question. We know the volume of the optical focus cuboid that we're looking at because we know it's 1 millimeter on a 1 millimeter microscope. We know it's 1 millimeter wide by 0.75 millimeters high by about 300 microns deep. And so we actually know what that volume is, and the resolution on the focus is quite good. So what we found is that we can make assumptions about that volume, and the volumetric accuracy of this when calibrated against a fluoroprobe is very good indeed. So the accuracy of the Bloom Optics app is 94%, which is about the same as you would expect from a lab.
And then we have a magnification question.
Ah, my favorite question. Yes. The short answer is the 1 millimeter is somewhere between times 200 and times 400, or if you're ignoring the eyepiece, that's times 20 and times 40. It's somewhere in that range, and it's subjective about where you think it is. The 2 millimeter has got a lower magnification than that. Digital microscopes are interesting things because, of course, I can project this digital image using a projector. I can project it onto a wall, and I can make it 20 feet across, and I can give you times 200,000 magnification very easily. And if you go on Amazon, you'll see digital microscopes advertised at times 1600, times 2000. But that's wrong because you have to pay a lot of money to get times 1000 under oil immersion in a lab. So there's something going wrong there. And the difference is the resolution, and that is why we quote the resolution. We quote this 1 micron resolution, which is roughly equivalent to a well set up times 200 microscope in a lab. But it's the resolution you need to be interested in, not just the magnification, because we can make things as big as you want. It's easy. Temperature from Amar about temperature, plus 40 C. Yeah, that's not a problem. So normal range. Basically, what we say is these are hand-operated temperature devices. So apart from the incubator application we talked about earlier, they're going to be in the same environment as you are. And if you're happy in that environment, then the microscope will be happy in that environment. So yeah, plus 40 is fine. Plus 50 is a little bit warm, and it's probably a little bit warm for you too. Minus 20 is fine, centigrade. As always with microscopes, you get problems below freezing with condensation and icing. And those are the problems, not whether the microscope works or not.
So just to, sorry, I just wanted to add one more point here. So if we want to keep these device outside for a longer period of time, for continuous monitoring point of view, I was just wondering, Andrew, if you have any comments on that, or you have done any demos or any?
Yeah, we have. The thing to worry about, Amar, is water. Just try, as long as you can keep them dry, it's fine. The best experience I can tell you about is the US FDA forestry department who use these in Alaska, and they're looking for spruce bud blight. And those of you who know Alaska know that it gets kind of cold up there from time to time. And they had real problems with our microscopes at first because they were taking them out of the truck and the condensation was just landing straight on the microscope, and the lens was just fogging over. But what they found is if they just went and had a cup of coffee and let the microscope acclimatize to the temperature, the condensation would clear and it was fine. So they've used them up in Alaska in some pretty cold temperatures, and that's not a problem.
Okay, thank you,
Andrew. And we've also used it in Antarctica as well.
Yeah.
So it's been
Greenland
in
as
the
well.
Arctic.
Yeah.
For snow algae.
So I've been up to 5,000 meters, so 1,500 feet at Mount Everest Base Camp. And please subscribe to our YouTube channel. There's some great links to videos there. A video that was made by National Geographic of the ioLight microscope that they took to Everest Base Camp. And they've been looking for a microscope to take up Everest for years. And because obviously you can't carry a compound microscope up Mount Everest, it's just too big. And so in, I think it was, 2019, they took our microscope up Mount Everest to look at protozoa under the ice. So that's a nice case study.
Andrew, do you have any examples you can give of the ioLight used in any other environmental monitoring organizations or situations?
Yeah, I mean, probably the most relevant one to this audience is there is a very nice water industry demonstration facility just outside of Atlanta, Georgia, on Lake Lanier, which is a big freshwater reservoir that has some serious HAB problems, and there's quite a big river basin system there, and they've got legal issues with water coming down the river from upstream states and then passing contamination to downstream states. And as you know, there's litigation between states on the passage of contaminated water. They have, that's an independent team. They've evaluated our microscope and the Bloom Optics app, and they are absolutely delighted with what they're getting. It's really good at detecting harmful algal blooms in that lake. They actually didn't have any blooms last season, but they were still able to detect some HABs because they were at a very low level. They were still detecting them with this system. So that's a good one. And again, Andrew and Patrick can give you details of that work, and there's a testimonial that they wrote it for us as well.
I know some people are asking about Bloom Optics right now. So Patrick's just put the link in about the webinar we had with Bloom Optics if you want to dive deeper there.
Hey Patrick, that's so that you get your commission and I don't get my commission on selling Eagle's product. That's me.
Well, what I wanted to have you speak to is how Bloom Optics and ioLight ended up working, or how the app came to be and developed, because it's a North American based
study. So Bloom Optics is a spin out of a major engineering company called Ramboll. Now, Ramboll Group is a Danish global company, huge company, and they build water treatment plants. And as part of their work building water treatment plants, they developed an AI technology for recognizing harmful algal blooms in the lakes for their customers. And in order to do this, they went out to market to find a small, lightweight, portable microscope that would generate the images for them to analyze. And they found our microscope. And they also found that we were easy to work with because we're roughly the same size company. Actually, we're a really big company compared with Bloom Optics. So we formed a partnership and we signed an agreement in 2022. And we now work very closely, increasingly closely, with Igor and the team at Bloom Optics. So they sell our microscopes. If you look at that link that, you send the link to the video. Okay. If you look at that, you'll see Igor from Bloom Optics using one of our microscopes, they always use our microscopes. And there's some questions there. Is Bloom Optics app a subscription service? Yes, it is. There's this annual subscription you pay, and you get a discount on the second subscription. So to open a subscription for your organization, there's a price, and then your second, third, fourth subsequent subscriptions are less expensive. And Patrick and Andrea are distributors for Bloom Optics, so they can tell you all about that.
And before you just to add to that too, Andrew, is the 2 mil. There's like a Bloom Optics starter kit, which is your 2 mm microscope, the AI app, and then you get a case with all the starter slides and pipettes and all of that, and that can be just purchased online on Bloom Optics website, which we've added as well. So that just tells you more about the kit. And then if you want to add extra users to your microscope, there's also, it talks about that as well. And then
there's a question from Cheryl there. Thank you, Cheryl. For now, Bloom Optics is great for freshwater HABs. Yes, that's correct. So freshwater cyanobacteria HABs. Marine HABs, the database is now being developed. Yes, and if you're interested in working with marine HABs, please come and talk to us, and we'll get you onto that program. We just need to generate thousands and thousands of images and have somebody tell us what is in those images. And once we tag those, we can generate a database of marine cyanobacteria. You wouldn't be the first one there. This project is already up and running and live and active. So, basically the more images we can get from the more different sources, the better. So we'd really appreciate your help with that.
Great. Any other, so we've listed the Bloom Optics website. Any other questions regarding microscopes, marine, cell imagers,
incubators, human kidneys?
Yes, yes. You forgot to mention your favorite line, Andrew, which is you could sit in the pub while you wait for.
Oh yeah, that's right. Yeah, that's a little story. Because when I was at university, which was a long time ago, the labs that grew cells, the research assistants who were usually young postdocs would have to sleep the night in the lab and watch the microscope to see when the cells had finished, when the batch had finished. And now we can transmit those images you saw. Those can be transmitted via Google Photos or any photo sharing app. That can go to your cell phone, and that means the postdoc can sit where postdocs like to be, which is in the pub, and watch the cells growing, and just go into the lab when they're cooked. So that's a huge benefit to postdocs.
Yeah, that's my favorite story. Good. Well, if anybody requires any more information, we have Cheryl's got another question for you, Andrew. What is the lifespan of regularly used ioLights in the field?
Cheryl, the answer to that is we don't know. We've only been selling them for 8 years, and they're all fine. So we've never had one die of old age. So 10 years, something like that. They last a long time. They're very robust. They're strong things. Our recommendation is it's like looking after your cell phone. Don't drop your cell phone, but if you do, it'll be fine. It will.
Especially if you're going through customs and they make you take it out of your bag and it drops off of the wheel.
That's my favorite. So I did that at Gatwick Airport. I was repacking my bag and I dropped a microscope. I thought, oh, that's interesting. I wonder how high that drop is, because there's an international standard for drop tests, which is a meter. And it turns out that the customs, I got a tape measure out and measured the height of the bench, and it was exactly 1 meter. So that was great. So it's passed that drop test. What I'd forgotten, of course, was that the security guy was watching me do this and got a bit excited that I was measuring up his security area. Wasn't worried about the microscope at all.
Too funny.
And awesome. It was fine after that drop.
Yeah, I'm pretty rough on mine too, and they're fine. We're fine. Yeah, they get tossed around quite a bit. Good. Anything else? Just like to thank you, everybody. Thanks, Andrew, for joining us and just taking a deeper dive on the technology. And any other questions, you can follow up with Andrew, myself, or Patrick. We'll send a recording of this out so you can share it, pass it along, rewatch it as many times as you like. And anything else? Does anybody have any other questions other than that? I know we have a few more minutes, but we'll just
I just wanted to thank you, Andrea, and for hosting this, and Patrick for having me on. It's really interesting. As you can tell, I love talking about this stuff, and really pleased to be here. And if anybody, you know, one of the great things one of the good things to come out of COVID is that I don't always have to get on an airplane and come and see you. We can do this by video. So if you want to take a deeper dive, then let's arrange a video with Patrick, Andrea, and me, and we very happily sit and talk through this with you. Although I do like getting on an airplane and coming and seeing you guys every now and then.
agreed. Thanks, Andrew. Good. Well, yes, thank you everybody for coming. Yes, Sarah, me too. Good. Thank you, everybody. Have a great day, and watch out for more webinars. Thank you.
Thank you, everybody. Thanks for coming.